Hello, welcome to World of Microbes. In Toadie's video, we will have an introduction of marine microbiology. The microscopic entities make up nearly /98% of the ocean's biomass and play critical roles in regulating the global climate, driving biogeochemical cycles, and generating over half of the world's oxygen. Microbes are everywhere, including the ocean. A single liter of seawater has about 1 billion bacteria and 10 billion viruses. They play many different roles in the marine environment, from being the base of the food chain to controlling much of the flow of marine energy and nutrients and being essential to the ocean's health. In fact, marine microbes can be the canary in the coal mine for the marine environment. Marine microbiology is the study of the microorganisms, bacteria, archaea, viruses, and microbial eukaryotes in the marine environment, including their biodiversity, ecology, and biogeochemistry, the science dealing with the relationship between the geochemistry of a given region and its flora and fauna, including the circulation of such elements as carbon and nitrogen between the environment and the cells of living organisms. More than 70% of the Earth is covered by ocean. When we think of life in the ocean, we often think of fishes and whales, life that is visible to the naked eye, but we may be astounded to learn that most life in the ocean is microbial life new. Microbes account for more than 90% of ocean biomass and constitute a hidden majority of life that flourishes in the sea. What is even more surprising is that much of this microbial life remains unknown because we cannot culture it in a test tube, and it is difficult to observe in nature. Studying marine microorganisms is difficult primarily because over 99% of these species cannot be cultured using standard laboratory methods. This, combined with the extreme pressure, vast scale, and complex chemical dependencies of ocean environments, makes research incredibly challenging. The core obstacles fall into four main categories. The unculturability crisis. Standard laboratory growth media often lack the specific trace nutrients, temperature, salinity, or low substrate, oligotrophic, conditions these microbes evolved with. As a result, they fail to thrive or reproduce in petri dishes. Extreme and remote environments. Many marine microbes live in hostile conditions like deep-sea hydrothermal vents. Gathering them requires specialized submersibles and equipment to survive freezing temperatures, total darkness, and crushing pressures. Complex ecosystem dependencies. Many marine microorganisms do not live in isolation. They rely on intricate symbiotic relationships with larger host organisms, like sponges or corals, or chemical cross-feeding between different microbes in a community. Rapid ecological imbalance. When taken to a lab, fast-growing species frequently multiply quickly and overwhelm the slow-growing ones, destroying the natural balance and cell-to-cell communication of the ecosystem. Marine microorganisms are defined by their habitat as the microorganisms living in a marine environment, that is, in the saltwater of a sea or ocean or the brackish water of a coastal estuary. A microorganism, or microbe, is any microscopic living organism, that is, any life form too small for the naked human eye to really see, needing a microscope. Microorganisms are very diverse. They can be single-celled or multicellular and include all bacteria and archaea and most protozoa, as well as some species of fungi, algae, and certain microscopic animals, such as rotifers and copepods. Many macroscopic animals and plants have microscopic juvenile stages. Some microbiologists also classify biologically active entities such as viruses and viroids as microorganisms, but others consider these as non-living. Marine microorganisms play a central role in the health and balance of marine ecosystems. These microorganisms are not only the primary producers in the ocean, converting sunlight into chemical energy, but they are also the engines of decomposition and nutrient recycling. In surface waters, microscopic algae like phytoplankton absorb carbon dioxide during photosynthesis, generating oxygen and providing an essential food source for higher organisms in the food web, including fish and marine mammals. In the deeper ocean, bacteria and archaea break down organic material, releasing nutrients and sustaining life even in the absence of sunlight. Microorganisms act as the primary engines for recycling elements. They regulate the global carbon, nitrogen, phosphorus, and sulfur cycles, directly influencing Earth's climate. Marine phytoplankton and cyanobacteria, like Prochlorocarpus, generate the majority of the oxygen in our atmosphere. Through photosynthesis, single-celled marine algae convert sunlight into energy, providing food for all higher trophic levels in the ocean. Specialized bacteria and fungi break down dead organic matter and pollutants, keeping marine ecosystems clean and cycling nutrients. Marine actinobacteriology is a specialized branch of microbiology studying actinobacteria that thrive in oceanic environments. Renowned for their complex metabolic machinery, these gram-positive, filamentous bacteria are the source of tilde 70% of naturally derived antibiotics and bioactive compounds currently used in clinical medicine. Marine actinobacteriology is one of the major emerging areas of research in tropics. Marine actinobacteria occur on the sediments and in water and also other biomass, mangrove, and substrates, animal. These organisms are gaining importance not only for their taxonomy and ecology but also for their unique metabolites and enzymes. Recent findings from culture-dependent and culture-independent methods have demonstrated that indigenous marine actinobacteria exist in the oceans and are widely distributed in different marine ecosystems. Now the question is why marine actinobacteria are so special? First reason is their unique habitats. They inhabit coastal sediments, deep-sea trenches, mangrove forests, and salt pans. Some live as symbionts within marine sponges, corals, and ascidians. Second reason is metabolic adaptability. To survive in extreme pressures, high salinity, and cold temperatures, these bacteria synthesize completely unique secondary metabolites not found in terrestrial species. Rare actinobacteria. While genera like Streptomyces are common, marine environments are treasure troves for rare, hard-to-cultivate lineages like Salinispora, Marinispora, and Verrucosispora. Marine microbiology studies microscopic life, like bacteria, archaea, viruses, and microalgae, that drives global biogeochemical cycles and produces approximately half of our oxygen. Inshore, coastal, and open ocean, pelagic, microbiology differ vastly in nutrient availability, species diversity, and environmental stresses. Inshore, coastal, microbiology. Inshore environments are dynamic, nutrient-rich, and heavily influenced by land and human activity. Nutrient levels. High concentrations of organic matter, runoff, and sediment resuspension. Microbial communities, dominated by copiotrophic, nutrient-loving, bacteria and flexible opportunistic taxa like the Rosobacter clade, which possess large genomes that allow them to rapidly consume various nutrients. Anthropogenic impact. Coastal microbes are highly vulnerable to human-induced pollution, agricultural runoff, and microplastics. This intense exposure often alters local community structures and can lead to harmful algal blooms or coral diseases, e.g., White syndrome. Open ocean microbiology. The open ocean is vast and largely oligotrophic, nutrient-poor, stretching far from a coast. Nutrient levels. Low in nutrients, requiring microbes to develop highly efficient, streamlined metabolisms. Microbial communities, dominated by specialized, free-living bacteria like the SAR11 clade, which is one of the most abundant organisms on the planet. In sunlit surface waters, photosynthetic cyanobacteria like Synechococcus and Prochlorocarpus are the primary drivers of carbon fixation. Vertical structure. As you go deeper, light disappears, the aphodic zone. Here, marine snow, detritus sinking from the surface, fuels heterotrophic communities, and chemosynthetic microbes thrive around deep-sea hydrothermal vents. Across both zones, specific microorganisms govern fundamental processes. The biological carbon pump. Viral shunts. The biological carbon pump, BCP, is the ocean's biologically driven process that transfers atmospheric carbon dioxide into the deep ocean interior and seafloor sediments. By drawing down an estimated 11 gigatons of carbon annually, this critical natural mechanism keeps atmospheric levels significantly lower than they would be otherwise, making it a primary regulator of Earth's climate. The biological pump operates through three primary mechanisms that link the sunlit surface waters to the dark ocean interior. Number 1, the gravitational pump. Phytoplankton fix dissolved into organic carbon via photosynthesis. When these organisms die, or when they are eaten by zooplankton that excrete fecal pellets, the carbon clumps into organic aggregates called marine snow. Driven by gravity, this material steadily sinks into the deep sea. The migrant pump. Millions of marine organisms engage in the Deal vertical migration, the largest daily animal migration on Earth. Creatures stay in the dark, deep ocean during the day, swim to the surface at night to feed on plankton, and return to the depths by dawn. They actively transport carbon downward by respiring and excreting at depth. The mixing pump. Physical ocean currents, winter mixing, and small-scale ocean eddies actively push dissolved organic matter, DOM, downward from the surface layer into the intermediate and deep ocean waters. The viral shunt is a fundamental ecological mechanism in which marine viruses infect and rupture microorganisms, like bacteria and phytoplankton, releasing their internal biomass as dissolved organic matter, DOM. This recycles nutrients back into the food web rather than passing them up to higher trophic levels. Instead of being eaten by larger predators, zooplankton and fish, carbon and nutrients from microbes are redirected via a loop. Bacteriophages, or simply phages, viruses that infect bacteria, are the dominant viruses in the ocean. Upon infection, phages cause their hapless bacterial hosts to burst open through a process known as viral lysis, thereby releasing nutrients and organic matter into the surrounding seawater. This phenomenon, known as viral shunting, diverts microbial biomass from secondary consumers in the food web, such as plankton and fish, and into the pool of dissolved organic matter that is primarily consumed by heterotrophic bacteria. When bacteria die and undergo decomposition, their organic matter has the potential to contribute to either the pool of particulate organic matter, POM, or dissolved organic matter, DOM. POM consists of complex structures and is not easily broken down by marine microbes. Consequently, it is often transported to the deeper parts of the ocean. However, DOM is more readily digestible for microbes, thus becoming incorporated into their biomass. As the microbial biomass in the ocean expands, it becomes a food source for organisms at higher trophic levels, including plankton, which in turn serve as prey for fish. But phages can also prey upon these microbes. It is estimated that phages kill about 10 to 20% of heterotrophic bacteria and 5 to 10% of autotrophic bacteria in the ocean daily, resulting in a significant release of carbon, nutrients, and other trace elements into the microbial food web. The dissolved organic matter, in turn, triggers a bacterial feast as the microbes eagerly consume the newly available nutrients and carbon, limiting their flow through higher trophic levels. Hence, viral lysis promotes bacterial respiration that retains carbon in the oceans instead of releasing them into the atmosphere. In this way, phages indirectly help sequester approximately 3 gigatons of carbon per year. Deep-sea microbiology studies microorganisms thriving in the ocean's extreme depths, typically below 1,000 meters. These microbes survive extreme cold, pitch darkness, and crushing hydrostatic pressure, driving global elemental cycles and forming the foundation of unique ecosystems like hydrothermal vents. Chemosynthesis. Unlike surface life powered by sunlight, deep-sea microbes, like those at black smokers, use chemosynthesis. They convert chemical compounds from the Earth's crust, such as hydrogen sulfide, into organic energy. Symbiosis. Many deep-sea animals, including giant tube worms and mussels, lack digestive tracts. They survive by hosting these chemosynthetic bacteria directly in their tissues to generate food. Piezo. These microbes are adapted to incredibly high hydrostatic pressures. Their cell membranes and proteins feature structural modifications that prevent them from being crushed or rendered inactive by pressure.